Welcome to this lecture on basics of neuroscience. Today, we're going to explore a fascinating and crucial process in the brain called long-term depression, or LTD. When we think about learning and memory, we usually imagine our brain making new, stronger connections. But what if I told you that actively weakening and erasing connections is just as important for your brain's health and your ability to learn? In this session, we will uncover exactly how and why the brain does this.
To understand this concept, let us start with a simple analogy. Imagine you are walking through a dense forest and you find a well-worn dirt path. If people stop using that path or only walk on it very rarely, the grass and weeds will slowly grow back until the path completely disappears. Your brain works in a very similar way when it comes to storing information and habits. When two neurons communicate weakly or out of sync, the connection between them actively becomes weaker over time. This fading of the path in your brain is the foundation of what we are studying today. Now that we have a mental picture of fading paths, let us define this formally. In neuroscience, this fading is called long-term depression, often abbreviated as LTD. It is formally defined as an activity-dependent, long-lasting decrease in synaptic strength. I want to be very clear here. It is important to note that this is not related to the psychological mood disorder of depression. In this context, the word depression simply means a literal depressing or lowering of the connection strength between brain cells. It is a physical, structural change at the synapse. So, how does this weakening actually happen at the cellular level? The process begins when a presynaptic neuron fires at a low frequency, typically around 1 to 5 hertz. This slow, repetitive firing causes only a very small, slow rise in calcium ions inside the postsynaptic neuron. This is a crucial detail because a massive influx of calcium would do the exact opposite and strengthen the synapse. Instead, this modest calcium increase activates specific enzymes called protein phosphatases. These enzymes are the key biological triggers that initiate the weakening of the synaptic connection. Following up on those enzymes, let us look at what protein phosphatases actually do. You can think of them as molecular scissors that remove phosphate groups from proteins. When they do this, they trigger a cellular process that physically removes AMPA receptors from the postsynaptic membrane, pulling them back inside the cell. AMPA receptors are the catcher's mitts for neurotransmitters. With fewer receptors available on the surface to catch neurotransmitters, the next time the first neuron fires, the second neuron will have a much weaker electrical response. The connection has been successfully depressed. Understanding the mechanism is great, but you might wonder why the brain would ever want to spend energy making its own connections weaker. Think about it. If our brains only had mechanisms to make connections stronger, our neural networks would quickly become saturated and overloaded. It would be exactly like a computer hard drive with no free space left. Long-term depression is absolutely essential for clearing out old, unused, or incorrect memories. By weakening these outdated pathways, LTD makes room for new information to be learned and stored efficiently. Beyond just clearing space, this weakening process has highly specific functions in certain brain areas. For example, in brain regions like the cerebellum, long-term depression is actually the primary mechanism for motor learning. When you are learning to ride a bike or play the piano, you make mistakes. LTD allows you to correct those mistakes by weakening the specific synapses that caused the wrong movement. Without this ability to weaken specific synapses, our brains would lose their flexibility, and we would be completely unable to adapt to new environments or unlearn bad habits. Now that we understand the biological and practical importance of long-term depression, we can look at how scientists study it quantitatively. To truly understand how synaptic strength changes, neuroscientists rely on mathematical models to simulate these biological processes. One of the most famous and widely used models is the BCM theory, named after its creators, Bienenstock, Cooper, and Monroe. This theory provides a beautiful mathematical framework to calculate the exact rate of change of synaptic weight based on the firing activity of the neurons involved. Let us look at their famous equation. Building on that mathematical model, here is the mathematical heart of the BCM theory. The formula calculates dW over dt, which represents the rate of change of the synaptic weight over time. If this rate is negative, we have long-term depression. Let us break down the symbols. The Greek letter eta is the learning rate, a small positive constant determining how fast the synapse can physically change. The variable x represents the presynaptic input activity, while y represents the postsynaptic output activity. Finally, theta m is the modification threshold. This is a critical value of postsynaptic activity that acts as a tipping point. When the postsynaptic activity y is greater than 0 but less than the threshold theta m, the term in the parentheses becomes negative, driving the entire equation negative and causing synaptic weakening.
Let us apply this formula with a concrete example to see how long-term depression is calculated. Imagine a synapse where the learning rate eta is 0.1. The presynaptic neuron fires with an activity level x of 2 units, and the postsynaptic neuron fires at y equals 5 units. The modification threshold theta m is set at 10 units. We want to find the rate of change. We substitute these known values directly into the BCM equation. First, we multiply the learning rate and the activity levels. 0.1 times 2 times 5 gives us 1.0. Next, we evaluate the threshold term. 5 minus 10 gives us negative 5. Multiplying these together, we get a final answer of negative 5.0. Because this rate is negative, it physically proves that the synaptic weight is actively decreasing, which is exactly long-term depression. Now, let us try working backwards to find the tipping point of a neuron. Suppose a neuroscientist observes a synapse where the presynaptic activity, x, is 4, and the postsynaptic activity, y, is 8. The learning rate, eta, is 0.05. The scientist observes the synaptic weight decreasing at a rate of negative 3.2. What is the modification threshold? We plug all these given values into the BCM formula. First, we multiply the constants on the right side. 0.05 times 4 times 8 simplifies to 1.6. Now we have negative 3.2 equals 1.6 times the quantity 8 minus theta m. We divide both sides by 1.6, which gives us negative 2 equals 8 minus theta m. Using basic algebra, we find that theta m equals 10. This means the neuron's threshold for switching between weakening and strengthening is 10 units of activity. Let us visualize this entire process with a graph of synaptic strength over time. On the vertical axis, we have the synaptic strength, measured as a percentage of its baseline. On the horizontal axis, we have time in minutes. Notice the dashed line at 100%? This represents the normal resting strength of the connection. For the first 15 minutes, the synapse is stable at this baseline. Then, at the 15-minute mark, low-frequency stimulation is applied to the neuron. Look at what happens to the solid curve immediately after. It drops sharply from 100% down to 60%, and it stays there. This sustained drop perfectly illustrates long-term depression. The low-frequency firing has caused a permanent, long-lasting decrease in how strongly these two neurons communicate.
We have covered a lot of ground today, so let us review the key takeaways. First, long-term depression is a fundamental mechanism of neuroplasticity that actively weakens synaptic connections over time. Second, we learned that it is primarily triggered by low-frequency stimulation, which causes a small calcium influx and leads to the physical removal of AMPA receptors. Third, this weakening is absolutely crucial. It prevents our neural networks from overloading and provides the flexibility needed for motor learning and for getting outdated information. Finally, we saw how the BCM theory mathematically predicts this depression whenever postsynaptic activity falls below a critical threshold. You now have a solid understanding of how your brain clears its pathways to keep you learning.